Okay, so I just wanted to go over how the microbial syntrophy lab worked and how the results can be interpreted again. So just a reminder, syntrophy or cross-feeding is the ability of an organism to utilize materials that have been excreted by other organisms. And we actually examine syntrophy in different mutants of Serratia that have lost the ability to synthesize its red pigment. And so seen here, these four different pathways are actually known mutants that we are working with. So the first one is actually going to be the wild type, and it's not shown on the plate here, but we know that the wild type is going to be a red color, okay? However, what we do not know is that which of these mutants belong to A, B, or C. Okay, so that's what we're trying to figure out here. Where do the mutations occur for each of these different strains? Okay, so just for ease of explanation, I'm going to refer to them as our first, our second, or our third strain, and we're going to ignore the wild type for now. Okay, so now we can go and look at what's the best way to tackle these enzymatic pathways? How do we interpret the metabolites that are produced? The easiest place to start, I feel, is to look at all the different enzymes that have been crossed out. So in the first strain, it can no longer make the second metabolite, so we're just going to cross that one out. The second strain cannot make the third metabolite, and the third strain cannot make the fourth metabolite. That means that here, that third strain is never going to be able to make the red color because the red color is, that red pigment is actually the fourth metabolite. Okay, so this implies that B is actually going to be the one that cannot make the red. Remember, it cannot make that red. So that implies that B is the strain that's here because it cannot make number 4 because you don't see any growth, any red pigment that's happening next to any of the neighbors for B. Okay, so that's like the easiest one to eliminate because again, it is missing that last final step to make the red pigment. Then we can start to look at number 2. So metabolite number 2 is missing in the first strain. However, it is being produced by the second and third strain, but in the second strain, it's not being converted into 3, so it's actually being diffused into the neighboring cells. Okay, so just to show that a little bit better, the 2 that's happening here in the second strain is being diffused into strain 1 and strain 3. Okay, so that means that the first strain can successfully take it all the way to 4 using that 2 to 3 to 4 from the second strain. And the third strain is already producing the 2 and it already can take it to 3, but it's just getting extra from that second strain. Okay, so that's what's happening. But we don't know exactly which strain, if it belongs to A or C yet. But when we look at metabolite number 3, we can see that 3 is being produced here by the third strain, and it's also being produced by the first strain, by the first strain. However, it's missing in the second strain. So that means that this metabolite number 3 from the third strain is going to be diffusing into the second strain, and it's also going to the first strain. Okay. So that means that the second strain, although it was missing the ability to make 3 of its own, it can use the 3 from the third strain and then continue the pathway to make the red color. Okay, but even though the first strain is producing 3, because it's able to take it through to make its own red pigment color, it is not being diffused out at all. That is not happening. And that's because it's using it for itself to make that red. Okay, so that means that because this second strain can only get the red color from a single neighbor, it's going to be strain A, which means that the last strain is going to be strain C. Okay, so in case that didn't make sense, we're going to look at it on the plate itself now. So in all of them, they all produce metabolite 1. Okay, that's just what we know. But then we can start to fill in the other metabolites. So B is going to be giving 3 to its neighbor. So it's giving it to C, and it's also giving it to A. Okay, that's based off of this diagram here. C is also producing 3, and it's converting it into 4 on its own. Okay, but it doesn't have the precursor. C is missing that precursor. It's missing 2, and so it's getting that from A. So 2 is being made by A and diffusing into here, and that 2 is actually what is going to here. So it's making the 2, that goes to the 3, that goes to the 4. Okay, but A is also producing 2, that's diffusing into B. Okay. However, don't forget, B can already make 2 on its own as well, and so it's making 2 right now. And then 1's going to 2, 2's going to 3, and it's making its own 2 to 3. Okay. And then just to finish the cycle, we have 3 that's going to 4 within C itself because it's getting that 3 extra from B, and so it's producing the red color on that side as well. And then in A, it's getting the 3, and then it's going to become 3 to 4 to produce that red color within A. And just to, as a reminder, we are not seeing any of the red color here. We're not seeing any red color there in A. So remember, there's no red that's happening right there because the 3 that's being produced by C, that 3 is going back into the 4 for itself. So it's staying contained within C. And so again, C is able to convert everything to the red pigment color on its own, or not on its own, but with the help of its neighbors. However, A and B are only able to do that partially so because B is not able to make the red pigment color at all, and A is only able to get that full pathway when it receives the third metabolite from B. So I hope that cleared up some confusion about this topic, and yeah, hope that helped.